Inflammatory bowel disease is an umbrella term for two conditions, Crohn's disease and ulcerative colitis. Crohn's disease is a condition of the small or the large bowel where segments of the bowel become inflamed and they don't work so well. Ulcerative colitis is a condition affecting the colon but not the small bowel. Inflammation is subtly different to that in Crohn's disease and the treatments can be different. This is a disease that is more common in younger people. It's more common in westernized countries. There's clearly an environmental factor associated with living in the West or with developed countries that is triggering IBD. Factors might include lifestyle and smoking. In addition, there may be a role in some patients for genes. We know by studying families that cluster with IBD that there are clear gene patterns that are predisposing them to developing IBD.
The only way of being able to make any difference in understanding the diseases and devising treatments is to assemble teams of clinicians, of scientists, mathematical modelers, and bioinformaticians.
We are a clinical partner, one of the 3 clinical partners in the SysmedIBD consortium. Our role is to provide patient samples and furthermore also to perform experiments on these samples.
In Liverpool we've recruited about 120 patients, but there have been patients involved from different centres in across Europe as well, in Aachen and in Maastricht in the Netherlands. They've recruited similar sorts of numbers to us, and those different cohorts being combined so that we've got information from people who come not just from Liverpool but from different parts of the European Union as well.
In SysmedIBD project, we try to get further insight in the exact details of this inflammatory process and hopefully find biomarkers or markers in blood or stool which can learn us why some patients have a very quiescent disease course and other patients have a very severe disease course.
Our company provides a software and databases, knowledge bases actually, to understand the disease mechanisms. We want to understand how disease is working.
TNF is one of the signaling molecules produced by cells that warns them against that there is an ongoing infection. And in IBD, immune cells produce too much of this molecule, so that leads to overall inflammation.
What we know is that there's a pathway, it's called NF-κB. When it's switched on, these cells produce inflammatory mediators, which tells the body you are inflamed. And we are studying this pathway because it has been noted that this pathway is not only switched on and off, it's oscillating. And this has been done in single cell analysis in the laboratory. And we are studying this now in tissues, in the whole body, and in patients. And we have learned that this oscillation also occurs in the patients, and there are some differences between normal patients and the patients who have inflammatory bowel disease.
We found a couple of interesting new compounds, chemical compounds, which are actually coming from those drugs which are already in the market, which has a promising effect on changing the control of the genes.
We try to understand changes in patients on an individual basis. And this comes then to the new concept of personalized medicine, so that we don't treat in the end a disease in the same way for each patient, but that we try to subclassify patients, that we see one patient is a little bit different from the other and needs maybe a different treatment.
We could identify patients which had a very mild disease course, and we could identify patients which had a more moderate or severe disease course. This sounds maybe very simple if I put it like that, but at this point it's very difficult to predict the disease course of a patient. And we now had the opportunity to better characterize this natural disease course and to go back to the patients to see if we can find factors which really predict this disease course.
Because we can now identify those people, we hope to be able to choose drugs early in their diseases that will control their disease effectively for them without needing to give extra toxic drugs to people who are going to have minimal problems in their lives.
We're just embarking on some exciting experiments that have arisen entirely out of the SysmedIBD project to repurpose a drug that we've known for 30 or 40 years that in the lab today seems to be working well in model systems, and we hope to take to patients in the near future.
You know, the Russian writer Leon Tolstoy says once that when people are happy, they are kind of similar to each other. But when they are unhappy, they are really unhappy in a different way. And that's what we found, that the unhappy people, the sufferers of IBD, they have a different mechanisms of their suffering. And if we really find the personalized treatment, so we can approach those personally and then make them all happy.